Hi everyone, welcome to our fourth submodule, writing about introduction and discussion. Oftentimes, sentences from these two sections are interchangeable, so always keep an open mind when writing these two. Most importantly, is we get the right flow and components in each section. Now, let us detail the introduction section. As you know, it acts as a prelude to our whole manuscript. Notably, readers who might not have expertise in the research area could appreciate and understand the context of the whole paper by reading this introduction section. This will also help to bridge scientists from different fields to integrate such research or ideas in their area of interest. What do you look for in an introduction? 3 elements are required as presented here. Is your research reported in this article new? Is it significant enough for scientific communities? And whether your manuscript is suitable for the journal? All of these must be answered within this section. Always remember these 3 when you start your introduction. In terms of structure, this section has 5 stages. First, we will introduce a general statement about a research field, including what is the big problem in that area. Then, we will describe previous studies that are trying to tackle the problem. Obviously, these studies may not have fully answered the problem yet, and that is why you need to do this research. This will lead you to the third stage, the gap in literature or in your research niche. Then, the purpose, objectives and/or main activities will be written, to further solidify our research framework and need. Lastly, we can justify the current work, but this is just an optional statement. You may find this last stage in a handful selected papers. Let us discuss each of these main stages, especially the first 4, next. In this first stage, we will introduce our field in a broad manner. We need to construct the right setting for the readers. For example, if your work is on transgenic rice capable of withstanding high temperature, you may start by telling readers about global warming and how it impacts food security. Then, go to your organism of interest, in this case, rice. Why is it so important to study rice? Having some statistics here will further increase the paper and research value. Please also remember to cite primary literatures properly in this stage and in fact, throughput the introduction section. When we say literature, it means all published research or review articles, books, websites which are peer-reviewed or from reputed organization. This is vital to inform readers that you know the extent of your work. Next, in the second and third stage, a more specific problem and hence gaps are introduced. Using selected literatures, we will build a gap in knowledge and a specific niche in our study. This is justifying why our current work is needed and hence merits publication. For instance, using our transgenic rice example earlier, the current gap in knowledge may be that our current approach using CRISPR technology is new, and the impact of our selected gene insertion or deletion has yet to be studied. This sets up the focus of our paper. Now, please always remember these signal words to mark your gap in literature. Use words such as not well understood, however, remains a challenge, rarely, needs further investigation, or presently unclear. These signal words are effective ways to highlight the research need and niche. Pay attention to them when you read any article of interest. This will tell you right away the motivation of the article and research. In stage 4 of the introduction section, we will detail the purpose, objectives and main activities reported in our manuscript. There are also several keywords for this section such as therefore, thus, hence. In this study, and this is the first study, etc. Use these words to lead you to the specific objectives and aims of the reported study. Although you can start writing this introduction section, wherever stage you like, here I offer you a suggestion. You may start writing up with the stage 4 statement first as it is one of the easiest, and will sit at the end of this section. Then move up to stage 3 to describe the need of this study leading to stage 4 earlier. Then, you can write about stage 1 to set up readers with background knowledge before completing stage 2, which may require the most extensive literature search. Now, let us do some quick exercise. Please go to your article of interest and identify these stages in the introduction. You may also compare with a few articles to give you a broad idea how this is done. In this next section, we will dissect on how to do the discussion section. I call this the end game, as this is where the battle where you decide the outcome of your experimental result. It involves deliberation of such results in the realm of relevant scientific area. What this means is that the results obtained must be situated and discussed together with current knowledge in literature. More readings are required, especially to explain the results obtained. First, let us consider the vital issue of its structure. Discussion, as you know from previous class, can be combined with result section. Conclusion section also sometimes embedded in here. The only way to know which structure to choose from is by inspecting journal specification, as well as articles recently published by that target journal. Discussion is also where the main theme of the paper is decided, and hence a direct relationship with the manuscript title later. As described earlier, discussion is also related to the introduction section. But remember to not regurgitate the information between these sections. There are of course several key messages and points we need to highlight in the discussion. First, we may start with the main purpose or hypothesis, or the main activities conducted. Next, we need to explain our current findings. How do they answer our research questions, hypothesis or objectives? Here you need to contrast with previous papers. Do they also get fairly similar result or completely different? If different, then why? This will lead us to the next point, which is the limitation of study. Is there any assumption here that we have taken to limit the result interpretation? Is our sample size large enough? Are there any other different populations that we need to sample to get a more representative result? Then, we need to discuss the implication of our findings in a wider context, and lastly, we need to recommend future research or practical applications. Let us consider an example, using our earlier scenario of transgenic rice. In this discussion, we will first start with the main experimentation conducted onto the rice, such as growing in higher temperature and other subsequent physiological and molecular studies. Then we will describe the result. Say for instance, indeed the yield of such transgenic rice is statistically higher than wild-type rice during high temperature growing. Then we can search in literature and cite those who may have conducted similar studies, but perhaps using different transgenic plants. Do they support our findings? If not, then why? Perhaps different transgene or plants have different regulation towards temperature stress. Then, we can detail the implication of our findings. Can this transgenic plant be introduced in field trials? Will this help to supply more rice in the future? Lastly, we may recommend future studies or applications, of which in this case, the possible adoption of these transgenic rice to various farmers for commercial production. Now, shall we go to our article of interest and see how these different elements fit in their discussion section? All the best, everyone.